All right. Hello, everyone, and welcome to today's cryo-EM Current Practices webinar. You're joining the three NIH-funded National Cryo-Electron Microscopy Centers. My name is Christina Zamani. I'm a scientist and training liaison at the National Center for Cryo-EM Access and Training, NCCAT. I'm joined today by staff members from all of the centers. Also here from NCCAT is Ed Eng. Joining from the Pacific Northwest Cryo-EM Center, PNCC, is Omar Devolku. And from the Stanford SLAC Cryo-EM Center are Nathan Burrows and Mike Schmidt. And each center is going to give a quick update on the resources that we make available at no cost to the research community. And then we'll turn the floor over to today's speaker, Rob Miller, who's joining us from MIT today. But I'll introduce him properly after our round of center updates. This webinar is a regular event. We host the last Thursday of each month, and we highlight users of the National Cryo-EM Centers who generously share the tips and tricks that have enabled them to make use of cryo-EM in their research. You can save the date already for our February talk on the 27th. We're still waiting to finalize the details, but once we have those, you'll be able to find the registration link on the events page at cryoemcenters.org. We'll also send it out on our regular mailing lists, so you will hear about it in a few places. Also, I'll point out on that cryoemcenters.org site, you can find recordings of past talks. One quick announcement, anyone who's planning to go to the Biophysical Society meeting next month in Los Angeles, if you want to come talk to us in person, we will have a booth in the exhibit hall, booth 623, and staff from the centers will be there, some maybe with posters and talks, so look for us, and if you can't find us otherwise, come find us in the exhibit hall. For final logistics today, if you do have questions for any of the center staff here on our panel, feel free to reach out to them using the chat at any point. But for questions for the talk, please send those using the Q&A box. You can send them at any point during the talk, but we will moderate Q&A at the end. All right, so let's get started. So first, I'm going to ask Omar to let us know what's new at PNCC.
Thanks, Christina. I am Omar Devolku, one of the microscopists here at the Pacific Northwest Cryo-EM Center in beautiful Portland, Oregon. Here at PNCC, we offer our users a seamless workflow from sample preparation all the way through to 3D reconstruction, including our screening access mechanism in which users come out and work with us face-to-face for a week through the entire workflow. As shown here, we have a number of microscopes equipped with different combinations of K3s, Falcon 4Is, Gatan energy filters, thermo energy filters, fringe-free imaging, and we offer in-person training for everything that we have. As Christina mentioned, if you have any questions, please reach out here or at our website, pncc.labworks.org.
Right, I'll turn over to Nathan to let us know what's happening at S2C2.
My name is Nathan Burrows. I'm one of our cryo specialists here at SLAC, and I manage our screening program, which is otherwise known as our exploratory screening and optimization program. And not only do we have that access program for our users, we also have a high-end data collection service program for our users who really struggle getting time on a Krios to collect their large data sets. We also have some exciting new program activity. We have a screening boot camp for advanced sample prep training, as well as a data processing study group, a data clinic that meets weekly as well. And we offer training programs tailored to your project, as well as an in-residence training program. We have the full suite of microscopes for Krioses, a Glacios, and a Tundra coming online. And we have, of course, our data facility as well to help us do all those computations. Feel free to contact us through our website if you have any questions.
Thanks, Nathan. Now I'll turn over to Ed.
Hi, I'm Ed, coming from NCCAT. Similar to the other centers, we offer high-end data collection, screening, and training, something that we're trying to work through is bringing training to your neck of the woods. So Christina, our training liaison, and Eugene, our screening lead, is trying to organize a workshop in Delaware. So if that's of interest to you, do ask us more. Something I do want to point out, it is conference season. You heard about BPS, but microscopy and microanalysis is going to be in Salt Lake City this July. Next week or in a couple of weeks is the deadline for papers. That's coming up February 14th. Unofficially, I hear in good order that the deadline will be extended to the following Tuesday. I want to point out there are three structural biology and biological sciences symposia that would be of interest. For example, B01, that's the 3D MFIG one. B07 is cryo-ET progress and potential, and B08, advances in cryo technology. So if you're going to be in Salt Lake City and you want to learn more about cryo, please think about going to M&M and submitting your paper by February 14th. If you want to find out more, go to ncat.nysbc.org.
Thank you, Ed. So now let's get to the highlight of today. Joining us today is Rob Miller. He is currently a postdoctoral associate at the Massachusetts Institute of Technology, working in new methods development for cryo-EM. Today, however, he's going to present work that he did as a graduate student at Cornell, where he used a solid background in physical chemistry and multiple complex experimental techniques to uncover new insights into biological processes, including one of my personal favorites, Allosteric. So Rob has taken some gorgeous snapshots of biomolecules, but outside of the lab, he also enjoys capturing macroscopic snapshots, particularly of landscapes. Something else I learned about Rob is he's an accomplished, formerly competitive snowboarder. So it seemed very natural that a cold weather loving photographer would take some time today to tell us a story that includes his cryo-EM journeys. So thank you, Rob, for joining us today, and take it away.
Awesome. Yeah, thank you so much for the introduction. Can everyone hear me well and see my slides?
Yes.
Yeah.
Great. Yeah, thank you so much for the introduction and the opportunity to talk today. I'm really excited to share some of the work that I did during my PhD in the lab for Professor Nozomi Ando at Cornell, specifically investigating the mechanism of allosteric regulation in an enzyme known as aspartate transcarbamoylase, or as we'll call it today, ATCase. And at the beginning, I just want to highlight that this is a really big story, and I won't be able to tell the entire thing today and involve lots of other techniques and really a massive collaboration throughout the lab. And I want to highlight those people that really made this story possible. So a fellow graduate student in the lab at the time, Mike Patterson, actually collected the biochemical data. I'll show a little bit of that today, as well as collected the crystallographic data, some of which with NETI, and built some of the atomic models. And then a postdoc in the lab, actually had a background in organic synthesis, was capable of synthesizing a small molecule that was really essential for the crystallography experiment. So it took this huge group of people in the lab. And as I mentioned, I won't be able to cover everything. So please, of course, ask questions at the end. But if this story is interesting to you, please check out the preprint that we recently published in bioRxiv. But without any further ado, a little bit of background on aspartate transcarbamoylase, or ATCase, before I jump into all of our fun results. Some of you may be familiar with this enzyme, as it is quite literally, excuse me, a textbook example of protein allostery, serving as a paradigm of allosteric regulation since the seminal work of Monod, Wyman, Chougeot back in the 1960s. But ATCase is not only important because it serves as this paradigm of allosteric regulation, but also because it has a critical biological function, the first committed step of pyrimidine biosynthesis in all kingdoms of life. And this is the condensation reaction of carbonyl phosphate and aspartate to form carbonyl aspartate, which eventually goes on to make UTP, which is converted into CTP. And it's known in a classical feedback inhibition loop that the final products of this pathway, the pyrimidines, are capable of binding ATCase and downregulating the enzymatic activity, so they slow the enzyme down. Maybe not surprisingly, it's known that a parallel pathway products the purines, and I will say historically really ATP has been characterized, but I'll argue today that it is the synergistic pairing of ATP and GTP, where very similarly to the pyrimidines, they've been shown that they are also capable of binding ATCase, but in contrast to the pyrimidines, they upregulate the enzymatic activity, so they speed the enzyme up. And in this way, ATCase is capable of responding to the intracellular concentrations of nucleotides within the cell. But because ATCase has long served as a model of allostery, it's been studied extensively for over 70 years. And our structural perspective of ATCase has largely come from crystallography, which in combination with other biochemical techniques has revealed that bacterial ATCases are comprised of two functional subunits. The first of which is a catalytic subunit, which form two catalytic trimers. I have one catalytic trimer shown here. Each catalytic monomer shown in a different shade of blue or white with its own active site. And I just want to point out two aspects of each catalytic monomer that we'll come back to later in the talk. The first is shown in the shade of purple, known as the 80s loop, and it is known to undergo a conformational change as it binds the first substrate, carboxyl phosphate, or CP. And the second is shown here in orange, is known as the 240s loop, which is known to be responsible for coordinating the second substrate, aspartate. And ATCase binds its substrates in this ordered fashion, first CP and then aspartate. The crystallography and these other biochemical techniques reveal that there's a second functional subunit, a regulatory subunit, which form regulatory dimers, three of them in total. And crystallography revealed that each regulatory monomer is capable of binding up to two allosteric nucleotides bridged by a magnesium ion. And in the full assembly of ATCase, we see that it forms a heterododecamer, so 12 subunits in total, with the two unique subunits, the catalytic and the regulatory. And we can see from a state that I'll reference for the rest of the talk as an enzymatically inactive tense or T state, this nomenclature coming from that work of Minot, Wyman, Chajot, that particularly if we rotate the enzyme on its side, that these catalytic trimers are sitting stacked right on top of each other, and they're bridged together by the three regulatory dimers. But due to the large, extensive characterization by crystallography, what researchers observed was that as you provide this enzymatically inactive tense state, both of its substrates, it undergoes a large conformational change as it transitions into what we'll call an enzymatically active relaxed or R state, where, so it says this T state binds both of its substrates. There's this large conformational expansion where the two catalytic trimers effectively separate from one another and the enzyme transitions into this relaxed R state. And due to the nature of crystallographic experiments, typically using bisubstrate analogs, it was thought that this transition from a T to an R state required the binding of both substrates. So the binding of the first substrate, CP, was not sufficient to drive this transition from a T to an R state. And although crystallography revealed a lot of details about what this enzymatically active R state looks like, for example, how substrates are coordinated, or how regulatory nucleotides are bound at the face of these regulatory dimers, there is disagreement between structural techniques on exactly what this R state looks like in solution. And we'll actually come back to this quite a few times throughout the talk. But due to this large body of both biochemical and structural work, this naturally led researchers to propose two primary hypotheses of allosteric regulation. And the first was a relatively intuitive proposal, which is that as these allosteric nucleotides bind ATCase, they must be directly altering the structure and thereby communicating their regulatory effect. But as crystallographers began to solve more and more structures of this enzymatically active R state in the presence of different combinations of nucleotides, they saw something very interesting, which was that regardless of the identity of the bound nucleotides at the face of these regulatory dimers, or if nucleotides were bound at all, that crystallographic models of the R state shared a fixed catalytic trimer distance as measured from the center of mass of each catalytic trimer of about 55 angstroms, so an expansion of about 55 angstroms. And so this naturally led researchers to propose a secondary model for allosteric regulation, which is that rather than directly altering the structure, what these allosteric nucleotides must be doing is modulating the relative populations of inactive T states and enzymatically active R states. For in the case of our downregulators, the pyrimidines, they must be shifting the population towards the T state, and our upregulators, the purines, must be shifting our population towards the enzymatically active R state. And although the exact structural role of these allosteric nucleotides had remained a mystery up until this point, it was well understood that they had a drastic effect on the overall biochemical behavior. In order to capture this under more physiological conditions, including neutral pH and physiological concentrations of magnesium, a fellow graduate student in the lab, Mike Patterson, collected biochemical assays where he first fixed the first substrate at high concentration. So he provided CP at a high concentration and slowly titrated in the second substrate, aspartate. And he was able to clearly demonstrate that the overall biochemical behavior of ATCase is strongly dependent on the identity of the bound nucleotides. And I think that this is a little bit more clear if we just sort of zoom into either side of the spectrum, our most downregulated state and our most upregulated state, where we can see in the case of our pyrimidines, our most downregulated state, the synergistic pairing of CTP and UTP, that not only is the maximal rate of reaction decreased, but overall, qualitatively, the biochemical behavior, which we can observe through the sigmoidal nature or through something known as the Hill coefficient, this NH term, which is indicating to us that the cooperativity of binding with respect to the second substrate is significantly higher. If we contrast this with our purine bound state, this ATP GTP bound state, where the kinetics become Michaelis-Menten-like qualitatively, and this Hill coefficient begins to approach 1, suggesting that cooperativity with respect to aspartate is effectively abolished. And what's more, we can see through something known as this half saturation value, we can think of effectively the concentration in which our enzyme, or half of our population of our enzyme, is turned on. We can see that under physiological concentrations of aspartate in an E. coli cell, which are about 4.2 millimolar at a maximum, that this pyrimidine bound state is effectively turned off. And in contrast, we can see that the synergistic pairing of ATP and GTP is significantly turned on underneath these physiological concentrations of aspartate. So this is suggesting that ATCase is modulating the enzymatic activity by effectively shifting ATCase into one of two regimes, one where the enzyme is significantly turned on and one where the enzyme is effectively turned off. And so the biochemical data clearly suggested that these nucleotides were playing a critical role in the enzymatic regulation. And although crystallography had provided us a lot of details, again, about how these substrates are bound or how regulatory nucleotides are coordinated at the base of those regulatory dimers, we wanted to use a combination of solution-based or solution-like structural techniques that could hopefully provide novel insight into the role of these allosteric nucleotides. And so for our study, we used a combination of a technique known as small angle X-ray scattering, or SAXS, as well as cryo-EM. And although my talk will primarily focus on the results that we obtained from cryo-EM, combining this with SAXS actually proved critical to our studies because for those of us that aren't as familiar with SAXS provides us structural information about the protein in solution, or about the average ensemble in solution. And so it is inherently a relatively low resolution structural technique. But again, it's telling us about what our protein looks like in a more physiological environment, a solution-based environment. And what's more is it allows us to rapidly perturb those solution conditions and monitor a conformational change as a function of that perturbation. For example, as you titrate in a ligand, you can monitor a conformational change and rapidly change these solution conditions. And so this is absolutely critical for us identifying each of the enzymatic states that we pursued at high resolution using cryo-EM, and I'll discuss today. Now, for those of us that aren't familiar with the technique in practice, how it works is we take highly purified proteins or particles and subject them to an incident X-ray flux. They then scatter these X-rays onto a two-dimensional detector. We would then integrate this pattern to obtain a one-dimensional intensity versus Q profile. And we can just think of Q in terms of length scales, but in reciprocal space. So really large values of Q are going to correspond to small length scales, and our smallest values of Q are going to correspond to the longest length scales. And I won't go through all of the detail of our SAXS analysis in this talk today, just to say that the raw scattering profile alone is rich with information and is telling us about what our protein looks like in solution. But of course, we wanted to combine this with a technique that would provide us high resolution information under solution-like conditions, at least as compared to a crystal lattice. And so, of course, we chose cryo-EM. And I won't fully introduce the technique today, but because I know people here are interested, I will point out that really a huge limitation in our experimental pipeline was actually identifying conditions in which ATCase was happy on the grid. It's a relatively stable protein in solution and relatively large for cryo-EM with an approximate molecular weight of about 300 kilodaltons and this sort of pseudo D3 symmetry, but it really, really hates cryo-EM grids. And so this was a huge challenge for us, where when we performed our initial screening, we purified ATCase, we brought it to our Arctica on campus and performed some preliminary screening. We were very excited that we saw these relatively high contrast particles, which to me look like coffee beans. So we call this the coffee bean view of ATCase, and people think maybe it's like hamburger buns, but I really like coffee. So we were really excited. We saw these great high contrast particles, but by eye, we seemingly only saw a projection along a single view of ATCase, and this was concerning. So we collected a small data set, and through iterative 2D classification, we were able to see that actually we were observing two projections within our images. The first corresponding to a projection along that coffee bean axis, and a second corresponding to this sort of mystery second 2D class that I couldn't readily identify. And so using just simple projection matching, we were able to determine again that this view, this primary 2D class corresponds to the single view along ATCase, this coffee bean axis, but the second class actually corresponded to free catalytic trimers in solution. ATCase was disassembling on the grid, and this suggested to us that ATCase was strongly interacting with the air-water interface, inducing this preferred orientation problem, as well as causing ATCase to dissociate on the grid. And of course, this was a huge problem because it prevented us from generating reliable reconstructions of the holo or the full assembly. But this dissociated class also represented close to 50% of our total particle stack. So it's a significant number of our particles were falling apart on the grid. And to combat this, I tried a huge different number of detergents and rapid plunge freezing devices. But in the end, the condition I found that worked for all enzymatic states was to actually coat the grids manually in a layer of graphene oxide using an approach described by the Nogales lab. And this gave us three benefits. By 2D classification, we saw no evidence of ATCase dissociation on the grid, so it removed that negative effect of ATCase dissociating. And it also significantly increased the orientational sampling of ATCase. So we're seeing many, many views of ATCase on the grid. And what's more is it significantly increased the particle density per image, which allowed us to obtain relatively moderately sized data sets that yielded really large particle stacks, which was really useful later on in the downstream processing. So this was a huge limitation at the beginning of the project, but upon determining a condition where we could isolate ATCase on the grid and keep it together as well as sample many orientations, we first wanted to start with our most simple state and ask, can we capture ATCase at high resolution using cryo-EM? And starting with our enzymatically inactive T state, so no substrates bound or no allosteric nucleotides bound, we brought our sample, we froze it on a microscope and screened it on our Arctica. And we also went to our SAXS beamline because a really powerful aspect of SAXS is it allows us to ask, has our high resolution model truly captured what our particle looks like in solution? And so for those of us that don't stare at SAXS curves all the time, I'll step through this plot one curve at a time, where we first took ATCase to the beamline and collected its scattering here shown in gray. And then, as I mentioned, this really powerful aspect of SAXS is it allows us to compare this experimental scattering to a high-resolution model. So in agreement with what previous researchers observed, we saw that when we generated the theoretical scattering from a crystallographic model, so we orientationally averaged and hydrated it, and then compared it to our experimental scattering. For those that don't stare at these curves all the time, I will say that this did an exceptionally good job at describing the scattering we see in solution. So this is suggesting that our high-resolution model is doing a good job at describing the overall ensemble we see in solution, which is why we're really excited that when we solved our first reconstruction of the T state via cryo-EM, that not only did it go to relatively high resolution, approximately 3 angstroms here, but it did a similarly good job at describing the scattering we see in solution. So this is really exciting to us, suggesting that cryo-EM will be a viable technique to pursue ATCase at high resolution. But as I mentioned at the beginning, what we're really interested in is what role do these allosteric nucleotides have in modulating enzymatic activity? And before we could probe or investigate those effects, we first needed to determine conditions where we could fully saturate that enzymatically active R state. And this is another powerful aspect of SAXS, where we could simply perform a ligand titration and monitor the conformational change across that titration. And I'll point out that the SAXS data that I'll show for the rest of my talk will be shown through something known as a crack heat representation, where all I did is I simply took the intensity, this one-dimensional intensity versus Q profile from the previous slide, and I multiplied the intensity by the square of the scattering vector. And what this does is it emphasizes mid-Q scattering features, which are particularly sensitive to distances on the length scale of interdomain distances. For example, the distance between 2 catalytic trimers. So we'll find that this is a super useful metric for monitoring conformational changes in ATCase. Where in the case of determining a condition that fully saturated the enzymatically active R state, we first saturated the enzyme in an analog for the second substrate, aspartate, known as succinate. So we provided the enzyme succinate in excess, and then we slowly titrated in the first native substrate, carbonyl phosphate, until we saw no further structural change. And again, SAXS then allowed us to identify the exact condition in which we should pursue at higher resolution, the exact solution conditions that yielded a fully saturated, enzymatically active R state. So we then took that condition, we collected this data actually at NCCAT, and we're able to obtain a really nice reconstruction of this enzymatically active R state. And right away, we noticed something really exciting, which was that when we compared our reconstruction of the R state to all crystallographic models, and I'll remind everyone that regardless of the identity of the bound nucleotides, or if nucleotides are bound at all, that crystallographic models share this sort of fixed distance between catalytic trimers of approximately 55 angstroms. So when we saw that our first reconstruction by a cryo-EM of the R state was far more expanded than the crystallographic models, we were really excited. It was expanded on the order of approximately 5 angstroms more. And so right away, we wanted to turn back and say, does this agree with the scattering that we see in solution? And we first began by performing theoretical modeling from the crystallographic models. And in agreement with what previous researchers saw, we saw that there was a systematic deviation from the theoretical scattering of these high-resolution crystallographic models. And because we're working in reciprocal space, the shift to higher Q is indicating that the crystallographic model is more compact than the ensemble that we see in solution, which is why we're really excited when we saw this unprecedented and exceptional agreement of the theoretical scattering from our model generated from cryo-EM in our solution scattering, suggesting that cryo-EM was already providing us novel insight into the structural consequences, or the structure of ATCase in solution, in this case, the structural consequences of substrate binding. But again, we really want to understand how these allosteric nucleotides modulate this enzymatically active R state, leading to the effects on enzymatic activity that we observe. And so once again, we turn back to the SAXS beamline because we had identified conditions that fully saturated the enzymatically active R state. We first saturated the enzyme in that state and then slowly began performing nucleotide titrations, first beginning with the purines, where again, we fixed the enzyme in the R state and we began slowly titrating in ATP. We then fixed ATP at 5 millimolar and began titrating in GTP. And what we saw from this Kratky representation is that the second maxima of this Kratky was shifting to the left. It was shifting to lower Q, indicating that overall the ensemble in solution, or our structural envelope, was becoming larger. The enzyme was expanding in solution. And in contrast to that, we saw in the case of the pyrimidines, beginning first with CTP and then CTP and UTP, that they had the opposite effect, that they were shifting the second maxima of the Kratky to a higher value of Q, indicating that the overall ensemble in solution was becoming more compact. And this is really exciting to us because it's indicating that these allosteric nucleotides are modulating either the conformational or the populations of enzymatically inactive T states and R states, or they're directly altering the quaternary structure of ATCase. And what's more is these ligand titrations provided us, once again, the exact solution conditions to pursue these different enzymatic states at high resolution, which is exactly what we did, first beginning with this R state bound to CTP and UTP, our most enzymatically downregulated state. So we took this condition, did a little bit of screening on our Arctica at Cornell, and then brought our grids to NCCAT. We were able to collect relatively large particle stacks. And upon curating this initial particle stack of the R state bound to CTP, I transferred the particles into Relion and performed 3D classification without alignment. And I will say this was really one of the most exciting days of all of graduate school for me, because what I observed, in contrast to the R state in the absence of any nucleotides, where I only saw 3D classes corresponding to a single quaternary conformation, I observed that in the presence of CTP and UTP that there's a mix of discrete quaternary conformations, where approximately 25% of our particle stack is corresponding to this enzymatically inactive and compact T state. And the other 75% of this our particle stack is corresponding to an enzymatically active R state that is expanded, but in a novel conformation. And I'll remind you all that the crystal structures share this fixed catalytic trimer distance of about 55 angstroms. And we observed in our reconstruction of the R state in the absence of nucleotides that it had a trimer distance of approximately 60 angstroms. And this was just, so exciting to me, because this suggested that both previously proposed hypotheses of allosteric regulation in ATCase may be true, that allosteric nucleotides are not only modulating the relative populations of inactive T states and active R states, but they're also directly altering the quaternary structure of these enzymatically active R states. But of course, for us, we always want to turn back and say, do our high-resolution models agree with what we see in solution via SAXS? And so using a really similar modeling approach that I did for the T and R states, we used a combination of models and found that the best description of our SAXS data came from a mixture of approximately 20% of these compacted T states and 80% of this novel R state. And this was really exciting because it's in super close agreement with the populations that we observed by a 3D classification. And again, suggesting that these nucleotides are modulating the enzymatic activity, both by modulating the quaternary structure of this active R state, as well as shifting the population back towards an inactive T state. And so upon observing this discrete heterogeneity, we, of course, wanted to investigate all the other nucleotide-bound states that we had identified via SAXS, first beginning with the remaining pyrimidine-bound state, this R state bound to CTP, and then following up with the R states bound to our purine, so ATP and GTP. And in the case of our enzymatically active or expanded R states, the only particle stacks in which we observe discrete heterogeneity, so a population of an R state in a T state, is in our pyrimidine-bound particle stacks, suggesting that our downregulators are shifting the population back towards an enzymatically inactive T state. But what's more is when we compared across all of our expanded enzymatically active R states, we noticed that the degree of structural expansion was a function of the identity of the bound nucleotides. And I think this is more clear if we look at the separation between the two catalytic trimers. The structural element that sits between those two catalytic trimers is actually this 240s loop that I had mentioned earlier on. It's responsible for binding the second substrate. And it is the structural element that sits between these two catalytic trimers. And we can see as we go across these different nucleotide-bound states that the degree of structural expansion as measured from this distance between the opposing 240s loops is expanding. Again, really strongly suggesting that these nucleotides are directly altering the quaternary structure of ATCase. But once again, we had to turn back to our SAXS data and ask, does this gradient of R state structures that we're observing agree with the differences in scattering that we see at the SAXS beamline? And again, in this unprecedented, really, really exciting agreement, we saw that our cryo-EM models did a really great job at describing the scattering we see in solution. So this is what's really, really exciting to us. And although we will come back to the consensus reconstructions we used to do these models, as they're provided us a lot of information about how these allosteric nucleotides are modulating the overall quaternary structure of ATCase, we were also curious not only what potential sources of discrete heterogeneity were present within our particle stacks, but what potential sources of continuous heterogeneity were present and what role they may have in the overall allosteric mechanism or enzymatic activity. And so in order to investigate this, we used a relatively straightforward approach known as 3D variability analysis, which very briefly, from a consensus reconstruction, attempts to map the variability of a particle stack onto an imposed linear trajectory. And what we observed for all enzymatic states is that they display a single primary continuous mode, the sort of molecular breathing-like motion, where the enzyme samples between a more compacted and more expanded state. And although I spent a lot of my PhD staring at these movies, as they're really rich in information, for example, you can see the correlated motions of key helices with the degree of expansion of the enzyme. Really, we were interested on an atomic level, what is driving these fluctuations or the difference in quaternary expansion as a function of the nucleotide-bound state. And what's more, a sort of limitation of these continuous heterogeneity approaches is that they map this motion onto an arbitrary reaction coordinate or a latent variable. And so in order to map this motion onto a real physical coordinate, I took this trajectory and discretized it into 20 unique subvolumes uniformly sampled across the trajectory, and then starting from a consensus model, refined 20 atomic models into each one of those subvolumes such that I could convert this latent variable into a real physical coordinate, which corresponded to the center of mass distance between the two catalytic trimers. We then repeated that for each enzymatic state. And what we saw, similar to our biochemical data, is that ATCase is effectively being shifted into one of two regimes in our conformational landscape, where we can see in the case of our enzymatic upregulators, our purines, that the enzyme is shifted upwards for this more expanded state. In the case of the pyrimidines, of course, in agreement with our consensus structure, their fluctuations are pushed down the reaction coordinate towards a more compacted structure. And what's really important to notice here is that there's no overlap between our upregulated distributions and our downregulated distributions, suggesting that ATCases or allosteric nucleotides are tuning ATCases enzymatic activity by effectively shifting its population to one side of this reaction coordinate. But because we had these nice consensus reconstructions and corresponding atomic models, we wanted to ask on an atomic level, what is driving the difference in expansion between these different states? And once again, sort of turning to either side of the spectrum of our states, our most enzymatically downregulated and compacted R state, our R state bound to CTP and UTP, and then comparing that to our most enzymatically upregulated state, an expanded state, the R state bound to ATP and GTP, we saw something really interesting when we investigated the regulatory domain. So we simply rotated a regulatory dimer on its side, and we're going to call this the face of the regulatory dimer, where allosteric nucleotides bind here. We saw in the case of pyrimidines, due to an N-terminal tail interaction conserved across a regulatory monomer, driven by the identity of these nucleotides, that the face of this regulatory dimer was essentially flat. And in contrast to that, we saw in the case of our purines, or in this case, ATP and GTP, once again, driven by the identity of these nucleotides, that there is this conserved domain swap N-terminal tail interaction that causes a bending at the face of the regulatory domain. So naturally, we wanted to ask, does the degree of bending at the face of the regulatory domain map or correlate across the degree of expansion that we see for each one of the enzymatic states that we've identified? And it's again a very exciting day in graduate school to see how well the small degree of bending at the face of the regulatory domain, again, driven by the identity of the bound nucleotides, maps across the overall quaternary expansion. And I think we can see this really clearly if we go from our most compacted state, the CTP UTP bound state, to our most expanded R state, the ATP GTP bound state, where again, we can see that these N-terminal tails, as they reach across the domains, they cause a small bending at the face of the regulatory domain. And this small motion propagates out through these arms known as zinc domains that hold on to the catalytic trimers and then modulate the entire quaternary structure of the entire assembly. And this is really quite amazing that these small conformational changes driven by the difference in identity of these different nucleotides is propagating across the entire structure to modulate the overall enzymatic activity of ATCase. But a really common theme in this project is that every exciting result sort of led naturally to another exciting question, which was that if these allosteric nucleotides are modulating the degree of expansion of our already opened R state, what role do they have in the transition from our compacted T state to our enzymatically active R state? And to gain some perspective to that, for the last time, we turned back to the SAXS beamline, and we did a relatively straightforward experiment where we first began by shooting our enzymatically inactive T state and then providing that T state its first substrate CP. And not much to our surprise, we saw virtually no conformational change. In agreement with what previous researchers proposed, that simply providing the first substrate to ATCase is not sufficient to drive this transition from a compacted T state to an expanded R state, which is why we're really surprised that when we provided the enzyme just ATP and GTP, it underwent a large conformational expansion on the order of a transition from a T to an R state. And we were even more surprised, we observed when we provided this ATP GTP bound state, its first substrate CP, it expanded even further. In fact, it expanded to our most expanded R state, this R state bound to ATP and GTP. We, of course, wanted to verify if any other nucleotide pair capable of inducing this structural transition in the absence of either substrate or just the presence of the first. And what we saw is that no other nucleotide pair was capable of inducing a conformational change, irregardless if the first substrate was bound. And so this is another really powerful aspect of SAXS. This has allowed us to not only identify a potentially novel conformation, but it also, once again, gave us the exact solution conditions in which to pursue this at high resolution. And because previous researchers thought that inducing a conformational expansion in the absence of both substrates was not possible, we, of course, wanted to get a high resolution look on exactly what the state bound to just the first substrate in ATP and GTP look like. So once again, we performed a little bit of screening on our Arctica and then sent our grids out to NCCAT and collected a relatively large particle stack. And I could tell even as the 2D classes were coming in that we indeed had solved an expanded R state-like conformation, which was much more clear as we were able to obtain our 3D reconstruction where of course, we saw that the enzyme was indeed expanded in the absence of both substrates. So in the case of this state, it's only bound to ATP and GTP at the face of these regulatory domains in its first substrate, carbonyl phosphate, but it is expanded in this enzymatically active R state. But before we even began building atomic models, I could also tell something, an additional aspect of this map that was unique, which was that these opposing 240s loops that we mentioned earlier on were seemingly in a novel conformation. And I think that's more clear if we look at this atomic model, or if we can compare our atomic model of this novel conformational state, what we can call a classic or canonical R state structure. Whereas I've shown before, in all previously published structures of ATCase, which rely on bisubstrate analogs or both substrates being bound, that these 240s loops sit parallel to one another in opposing catalytic trimers and stacked on top of one another. But what we observe in this novel conformational state is that these 240s loops form a sort of head-on conformation where they're removed from the active site, which sits down here, and presumably allowing for the diffusion of the second substrate, aspartate, or substrate analog for diffusion into the active site. So this was really exciting. This combination of SAXS and cryo-EM, not only it allowed us to identify a novel conformational transition, but to pursue it at high resolution and even build in de novo novel structural elements not observed in other enzymatically active R states. But what's more is this atomic model allowed us to ask, what is preventing the other nucleotide-bound states from sampling this novel conformation in the only in the presence of the first substrate? And so to gain some insight into that, what we did is we extracted the catalytic trimers from our novel conformational state down to CP and ATP and GTP. We then docked those catalytic trimers into our most compacted R state model, the R state bound to CTP and UTP. And right away, it was very clear why these other nucleotide-bound states are incapable of sampling in this novel conformation, where particularly for our most compacted states, there's strong steric clashes between the opposing 240s loops. But what's more is that these 240s loops are relatively negatively charged, so there'd be a strong charge-charge repulsion, which then explains why ATP and GTP are the only pair of nucleotides capable of expanding the enzyme enough to relieve these steric and charge-charge repulsion interactions, allowing ATCase to sample this novel conformation. And then we really think that this novel conformation, this tuning of the overall expansion of the R state explains the biochemical behavior we observe, where in the presence of pyrimidines, the enzyme must cycle from an inactive T state to an enzymatically active R state in order to complete its turnover. Whereas in the case of ATP and GTP, the enzyme is capable of expanding to an enzymatically active R state prior to binding both substrates. And there's really this large combination of different techniques relying on SAXS and cryo-EM, as well as a lot of crystallography and biochemical data I didn't have time to share today, that allowed us to propose a novel mechanism of allosteric communication in ATCase, which is that for all nucleotide-bound pairs aside from ATP and GTP, that ATCase must reside in this enzymatically inactive and compact T state prior to binding the second substrate, which upon binding transitions to this enzymatically active R state such that it can complete its catalysis. Whereas in the case of this novel pair of nucleotides we identified, ATP and GTP, they're capable of bending the face of the regulatory dimer enough such as to relieve these charge-charge and steric repulsions caused by a head-on 240s loop conformation, such that the enzyme can sit posed, ready and active in an open conformation upon binding the second substrate, which then would only require the movement of these 240s loops into the active site in order for it to complete catalysis. And with that, I want to thank everyone in the ANDA lab. It's just a wonderful place to get a PhD, and everyone in the lab is incredible. But I really want to thank everyone on Team ATCase, so of course Nozomi, as well as Mike, who did lots of model building, crystallography, biochemical data, and many, many late nights at the SAXS Beamline with me, as well as Nettie, Xiao Quinn, Raleigh, and just everyone in the lab. I also want to thank everyone at CHESS, so my co-advisor Richard, as well as all the Beamline scientists that made collecting the data possible, everyone at the CCMR at Cornell who taught me how to use a microscope, how to screen, and of course, everyone at NCCAP are the same, and allowing us to collect really beautiful data that allowed us to put the story together. And with that, I would happily take any questions.
Thank you, Rob. That was absolutely fantastic. You're obviously not hearing the applause that I'm sure is coming from the crowd. We have a couple of questions that have come in already, and everyone can send in more questions now while we start looking at those ones. We'll start with a question from Dennis Winston, who asked, did you see any 3D classes with C1 symmetry that were missing regulatory subunits?
Yeah, that's a great question. So for all of the T-state reconstructions that we did, they typically represented a smaller fraction of the particle stack, so we had to enforce C3 symmetry to get reliable reconstructions. But for all of the R-state models, we did not impose, aside from this state, actually, we did not impose symmetry. And you certainly see a huge amount of variability across the regulatory domains in terms of overall resolution. And this could be due to simple alignment. It has a hard time aligning the individual domains. It could be because there's still some interaction with the air-water interface, which is causing some denaturation. Or they also could just be highly dynamic, which is what we're observing. So you certainly see quite a bit of variability from regulatory dimer to regulatory dimer. Oh, but never missing entirely, sorry. Never entirely missing, just variable quality, particularly where the regulatory nucleotides are found. That quality of that resolution is quite variable from domain to domain.
Nice, thanks. Then there was a question asking if you could clarify what you're comparing. I think actually I took a note. You were around slide 37 when this question came in. If you're comparing small angle X-ray scattering and small angle electron scattering, or what your comparison is to the small angle X-ray scattering there.
So in the case of this model, really what we're doing is SAXS is providing us overall structural envelope of our average ensemble in solution. So it's telling us on average what our protein looks like in solution. And so really how this is done in practice is not directly from cryo-EM maps. We build in atomic models, and then you generate the theoretical scattering from those models by orientationally averaging them in 3D space and hydrating them. There's many different programs that use different implementations of hydration layers or how they actually calculate the scattering from an atomic model. But we're directly comparing from a 3D coordinate file, basically, to the observed experimental scattering to try and gain some insight into, have we imparted some sort of artifact by our high resolution model or captured a state that doesn't represent the actual conformational space that we see in solution? So if we biased it towards a single structure that doesn't represent what we see in solution, or in the case of for ATCase and the crystal structures, are we compacting the structure due to the crystal lattice?
Great, thanks. Hopefully that clarified. If it didn't, the person who sent that question could send in a follow-up if you need to.
Yes. Yes.
Yeah. And then a number of comments coming in that say, great seminar, very nice work. So you guys are being very nice to our speakers. So I'm going to hit you with a couple more questions.
Great.
Actually, so you collected enough data. You probably got a sense of like how much data from each grid you would need to then do the downstream processing you needed to do. So I'm just curious, like how much data did you need from each condition?
Yeah, that's a good question. I think it'd be hard to put an exact number on it. I will say, I tried to collect relatively uniform size particle stacks. It's hard to do that exactly for each enzymatic states, but particularly, for example, this R state bound to CTP and UTP, when we begin to observe discrete heterogeneity, the larger the particle stack made a huge difference for our reconstructions, because inherently in the filtering process, when you're doing this 3D classification, there are particles that don't align well to either class. So you're losing particles in the 3D classification and inherently shrinking the size of your effective particle stack for each state because you're separating them into discrete states. So at NCCAD, on average, we would collect, I would try to do like 24 hours of data collection, which gave us close to 10,000 images. And because we had relatively high particle density, I would say on average, after curation, the particle stacks were on the order of 750,000 to 1 million particles. And the discrete heterogeneity as well as the 3D variability analysis really benefited from having larger particle stacks. You can notice a big difference between that and our screening data sets.
Thank you. All right. So, Andre Bulva says, very cool Allosteric story. The mechanism is relatively similar to the regulation of bacterial purine metabolism enzyme inosine 5' monophosphate dehydrogenase. The similar mode of binding of the pair of purine nucleotides in the regulatory domain in that enzyme allows for binding of dinucleoside phosphates. Like AP4A, AP5G, etc. Have you tried any of those signaling molecules with ATCase? Any other types of nucleotides or things like PPP, GPP?
So in our studies, we didn't systematically test like a huge library of different nucleotides. There is evidence that without the triphosphate, the magnesium actually coordinates between the third phosphate between each opposing nucleotide. So the absence of that third phosphate group makes it very difficult to coordinate two nucleotides per regulatory monomer. But we didn't like systematically test different effectors or nucleotide pairs in that sense. Although there is evidence that, for example, if you provide it ADP, that you won't see coordination in the face of the regulatory domains.
All right. There's a question about how well are the models refined at this stage, and is it useful to compare them to crystallographic structures?
Yes, that's a great question. So particularly for the regulatory domains, and I didn't have time to go into all this. There's a couple of beautiful figures in our paper about this, that crystallography was absolutely critical. Not only for having great reference starting models that are very similar, you can start with rigid body of individual chains and refine from there, but particularly at the face of the regulatory domains. And I'm not sure I have a great image of it here. I might have used, let me zoom in over here. That crystallography allowed us to really pinpoint exactly how these nucleotides were coordinated at the face of the regulatory domain. So for example, here is an unsharpened map, simply because the N-terminal tail density was extremely hard to resolve in some of our cryo-EM maps, which I think is a combination of alignment as well as just simple disorder, that they're moving around a lot. And so crystallography, I don't have a picture of the density here, but it was able to obtain super high resolution details, not only about how these N-terminal tails interact across the regulatory dimers for each nucleotide bound state, but it allowed us to confidently assign the identity of these nucleotides, which from the EM maps alone is very, very challenging to do. But with really high resolution crystallographic models, we were able to confidently assign the position of both ATP and GTP in this case. Thank you, Mike and Nettie. So yeah, it's really the, ATCase was, or excuse me, cryo-EM was able to capture these large conformational changes that really is from the crystal lattice. We're just preventing ATCase from sampling. It's squishing it down, keeping it at that fixed distance. But these really high resolution details, we absolutely needed crystallography for. Yeah, and I think that's a great theme of this project, is that combination of all the techniques really to make it come together.
All All right, here's a question from Sumakata. They're asking the nucleotide titrations with SAXS were impressive. Does the zinc binding motif have a role in the 80 loop in the T state?
Oh yeah, that's a really great question. I might have an image of that. So the zinc domain, and maybe it's just most obvious from this movie. The zinc domains are in very close coordination with this 80s loop. Now, the exact role of whether they stabilize this 80s loop is not very well understood because in the absence of the first substrate CP, this 80s loop is significantly disordered. There's no real structures that have resolved to great resolution exactly what the conformation of this 80s loop is in the absence of the substrate. So it's not exactly known the role of whether these zinc domains are stabilizing that domain in the absence of the first substrate. But upon coordinating CP, all of the contacts for the 80s loops, it's a lysine that dives in. They're coordinated by the substrate and the surrounding active site residues. That's what stabilizes the loop in the active site.
All right, I've got another mechanism question for you, and then we'll come back to a cryo-EM question. But there's a question about the changes of the cooperativity, and are there any explanations provided by your model?
Yeah, so there are definitely some follow-up studies, I think actually trying to capture ATCase under turnover conditions by a cryo-EM, which I did try and was very challenging to do, may give us some insight into that. But we think that it's actually this novel conformational state, this sort of head-on conformation that the most expanded states can sample that explains the biochemical behavior that we see. So, for example, in the case of our most downregulated state, I think this just makes it most clear. In the case of CTP and UTP, that the reason we see this cooperativity with respect to aspartate is that as the enzyme completes its turnover, it spits out product, it must collapse back down to the T state, then bind both substrates, undergo this large conformational change, complete its turnover cycle, and continue this over and over. And this expansion from a T to the R state must be coordinated across the entire enzyme, where all of the active sites were at least a significant fraction or a required fraction of them are bound before it undergoes this conformational transition. So that inherently imposes cooperativity, where in a state where the enzyme can sample an open conformation independent of if the substrates are bound, this is what allow the active sites to behave more independently because it's only local structural changes required in order to complete catalysis rather than huge quaternary structural changes which require coordination across the entire enzyme.
Great, thanks. Rob, if you have a couple minutes to stay, we may run after the hour, but there's a couple good cryo-EM questions here, some
Of course.
data processing questions. For those of you who do have to run off, we will still record, and that recording will be posted. And the national centers are here to help you collect lots and lots of data if you need it for your cryo endeavors. So please join us again. But we'll get right back to those last questions. Did you search for asymmetric conformations with symmetry expansion prior to 3D classification or do any symmetry relaxation? I wonder if poorly resolved NTDs might become more visible.
Yeah. So all of the final consensus reconstructions came out of RELION, where we systematically tested C3 symmetry relaxation. And quantitatively, you don't really see a drastic difference in estimated resolution, but qualitatively, you see slight improvement in exactly like nucleotide binding regions or regulatory domain in general, but not a great enough improvement in resolution where we could de novo assign nucleotide density. We still had to turn back to our crystallographic data to say for sure this is an ATP or for sure this is a GTP.
Thanks. And then there's a question about your 3D VA. So could you comment if there were tricks involved in your use of 3D VA or 3D classification, or if you had any particular struggles. I think that's a big question. So
Yeah. I should have mentioned that actually. So I will say the 3D classification in CryoSpark has undergone lots of updates, so it may be different now. But at the time when I was doing this, 3D classification, no matter what I tried using template inputs, and I even tried after knowing what the answer should be, going back into CryoSpark and providing it input templates, it was never able to capture this discrete heterogeneity. It would only report this R state, this expanded R state. We can never capture the compacted T state. Now, within 3D classification, I sort of systematically or empirically tested a filter resolution such that small differences in the expansion weren't separated into unique classes. Otherwise, it will begin to sort of arbitrarily separate out the different R states based on how expanded they are, which then just dilutes all of the classes. So I believe it was a 10 angstrom filter that I used, but I just low pass filtered the maps before performing the 3D classification. But really, I was only able to see that in Relion. For 3D variability was relatively straightforward. The only difference is so that we could look at any potential sources of asymmetry as I transferred the particles back from Relion, and I actually tried to refine ATCase in the highest possible symmetry operator, which is either C3 or D3. Sometimes not all of the particle stacks could be forced into D3, but upon enforcing that symmetry, we then performed a symmetry expansion, and the symmetry expanded particle stack is what we subjected to 3D variability analysis. We do see slight asymmetries. I can't speak necessarily to their functionality, but you'll see that like the distance between each one of the opposing catalytic trimers as measured from these 240 loops is actually not uniform across the trajectory. Certain of them are slightly more expanded and they're not perfectly in sync. This could be poor alignment across the trajectory, or it could be inherent real biological asymmetry. It's very hard to say from the trajectories alone.
awesome, thanks. All right, and then one last question for you. So are you saying that it's faster to collect and calculate SAXS data compared to cryo-EM data? So
Oh,
maybe more generally, like,
why
yeah,
use SAXS?
yeah,
Basically, yeah, that's a
yeah.
great question. So I could give a whole another hour on why SAXS is such an amazing and powerful technique. But really, for our study, this allowed us to rapidly perturb solution conditions. So if we would have been fishing for different conditions where we fully saturated in an ATP, GTP bound state, or just an ATP bound state, we would have spent years trying to freeze different conditions on a grid. And then we wouldn't have a source of some sort of ground truth measurement if we fully saturated a given state. So you would inherently see more structural heterogeneity if you only fully or half saturate a state. And so SAXS is really powerful for that alone, is that it allows us to really, within a period of an hour or two, you can perform a ligand titration and map an entire transition between two states and know the exact solution conditions. But SAXS is also just extremely sensitive to how happy your protein is in solution. Even a small fraction of protein aggregate or large oligomeric formation is extremely detectable by a SAXS as the signal goes with the square of the molecular weight. So SAXS is a way not only to monitor these conformational chains, but to ask an even more basic question is my protein happy in solution? And then you can rapidly perturb those conditions, change the pH, salt levels, and see, is my protein now more soluble? So as a starting point, SAXS is great just to see if your protein is happy. But similar like to the question that was asked earlier on, if we had tried other pairs of nucleotides, SAXS would be the perfect place to do something like that, where you could bring a library of different effector molecules, titrate them into your R state, and see what happens. And if nothing happens, then it's not worth spending a week on the microscope or lots and lots of money. Instead, now we can isolate, okay, these three states showed a clear conformational response to these ligands. Let's pursue them at high resolution. And then, yeah, there's lots more. The fact that we can verify high resolution models to really what our protein looks like in a solution, that gives us great confidence that we've truly captured the physiological assembly or consequence of these nucleotides binding.
If there's time, I'd like to ask a question or two. This is Mike. Hi. is a great, absolutely great seminar. And
Thank you.
since I started with graduate school a long, long time ago, allosteria has been argued over, and this is a great exposition of that thing with that particular enzyme, which I remember studying back then.
Yes, yes, yes.
So two things. Is there a state that you can generate which is balanced between 50% one form and 50% another form, and collecting data on that form, which presumably has the most equal balance between the two states, lets you explore the peak, the transition state between those two forms, and perhaps get enough examples along the transition that would say, oh, here's a stereochemically reasonable. Of course, you have to do the molecular dynamics along with it. Stereochemically reasonable pathway that gets you from one to another, and the halfway point, 50-50 mixture of the two, is the perfect place to actually do that.
Yes, that's a great question, and really why, like, sort of towards the end, I tried to capture ATCase under turnover, because so for the transition from the T to the R state, when we just performed, you could imagine selecting a point in between here where you sort of have half T state, half R state. But what I don't have shown here is we actually use a singular value decomposition in an assumption in SAXS analysis is that we're working in a so-called infinitely dilute regime, meaning that all of the scattering profiles that we see add linearly. They're just linear combinations of one another. And what we could show for these transitions is that they are a two-state transition. There is a population that's described by the T state and a population that's described from the R state. So this transition from the T to the R is very fast and concerted. The population shifts from T to R. And so watching that transition between the two states, in fact, I could send you a really cool MD paper where people use SAXS-driven MD to try to map this transition from the T to the R state. But to obtain actual experimental observations of that expansion, I think we would have to capture the enzyme under turnover. The issue is that it's very fast. So you'd have to super saturate the, but in that case, especially for states, aside from the ATP and GTP bound state, they presumably have to transition from the T to the R state every time they complete their turnover cycle. So if you can capture this mixture of populations, you'd have a lot of them that are collapsed and a lot of them that are already expanded, but you'd have some subpopulation that were in that intermediate. They were sampling the transition from the T to the R state. Now, I don't know if you could enrich that population enough to really get reliable reconstructions along that trajectory, but I think it's a really exciting, somebody should try.
Yeah. The inherent thing is that the transition state has the highest energy, therefore the lowest population.
Right. And
another thing is in terms of quantitating your fit to the atomic models to your densities, especially where you have states which are have inherent disorder or a mixture of states in there. One thing that actually quantitates it is the Q score, which actually says, okay, this is the match of your particular model, whatever model you choose, to the particular map that you're trying to fit it to, and that tells you, well, I can see this state, these atoms very well, and these atoms I don't see very well, and you can where those atoms are and where the heterogeneity lies.
Yeah, so for our consensus reconstructions, we didn't do like a sort of ensemble modeling to try and look at like different states of potential models, although that would be really interesting to do. Really, for assigning like reliability of assigning positions, we were lucky in the sense that for the core of the catalytic domains, they went to relatively high resolution, 2.2, 2.3. So building those was relatively straightforward. For the regulatory domains, like you said, there's potentially multiple conformations that could be sampled, and certainly certain residues that are well coordinated. For example, to the allosteric nucleotides, we can assign them with great confidence, but many of the residues, it becomes very hard to assign them with great confidence. And in that case, we typically relied on our crystallographic models and a little bit of biochemical insight to say, even though the structure is slightly different in the crystal structure, does this contact still make sense if we're missing the density? And that's sort of how we went through iteratively to build the lower resolution parts of the EM maps.
Mhm.
But that idea of trying to assign a score or build an ensemble, I think would be really interesting.
Okay. Thank you.
Nice. Well, thank you, Rob. And a number of
Yes,
comments came in like during the Q&A saying, thanks for the clear talk. This was wonderful. So you took us on a complicated journey, but very understandable one. So we really appreciate it.
thank
It's
you.
super fantastic.
Well, yeah, and thank you, you guys. Truly, NCCAT we didn't even have a cryo set at Cornell, so that in and of itself, but got to learn a lot, and yeah, I'm really grateful for the opportunity.